Thank you, everyone, for coming. My name is Brian Neiman. I'm the founder and CEO of Sanguine, and I thank you again for coming. Uh, I wanted to quickly introduce the company before we introduce our speaker today, Shruthi. And a little bit about Sanguine. I started the company about 10 years ago with the idea of working directly with patients. And by working directly with patients, we found that it was easier to collect samples and specimen information and the actual specimen itself, as well as medical record information and other patient-reported outcomes. And because of that direct-to-patient relationship, it's yielded for researchers like yourselves higher data coverage per patient, longitudinal information, as well as faster sample and data acquisition. Again, because of the direct relationship and the strength of relationships that we have. So the best way to think about us is that we are a platform connecting both researchers and patients to accelerate the research process through a better data and more data coverage per individual. And I'm happy to be here. Historically, over the past 10 years, we have completed over 600 research studies across autoimmune conditions, oncology, and rare diseases. And we're happy to introduce Shruthi Bharadwaj, excuse me. Today, her experience is in IBD and understanding the onset of colon cancer. And Sanguine, historically, as I mentioned, has been involved in specimen collection and the specimen, but now, as we're getting more into medical record data collection, we're seeing the use in HEOR, in patient-reported outcomes. We've also seen that use in understanding claims and pricing strategy, and now in artificial intelligence and machine learning. So with that, we're entering that part of the market and seeing value of medical record data in that regard. So I would like to introduce Shruthi. As I mentioned, she's focusing on IBD and colon cancer. She's a senior scientist at Novartis in AI in drug development, and she has a patent in machine learning and her approach to, as I just mentioned, colon cancer and IBD. She's won several NIH grants and has published several abstracts and book chapters. So we're really excited to have Shruthi here to talk about her work and how she's making an impact in IBD and colon cancer. So without further ado, Shruthi.
Thank you so much. Okay, before I begin, just a quick show of hands. How many are new to AI ML? Okay, a few. How many are already working on AI and machine learning concepts? Okay, for those of you, this might be a little not too exciting, but please stop me. For people who are new to this, stop me, ask questions. I think that's how we can get this forward. Like I was telling Raksha a couple of minutes ago, stop me whenever you have any questions. So like the introduction said, so I am working at Novartis. I work with a small team which focuses on AI and machine learning and using these approaches to clinical trial data analysis. Now, before I start, what exactly is healthcare data? Now, I'm again not completely sure about how many of you are already working with healthcare data, how much, how little, so I'll just start with the most basic thing. So from my experience, healthcare data combines a lot of different things. It's not just genomic or proteomic, but it's a combination of everything. So EMR is a big one these days, and you need to have a complete picture of the patient before you try to analyze anything. That is what we call precision medicine. That is where you have the EMR data, imaging data. The EDC essentially is, these days you have a lot of digitization happening. So you have these laptops, you have apps that capture information. So all these things combined gives us a picture of what the patient looks like. And that is the first stage in analyzing any kind of data, getting the data. Now, the reason AI and machine learning has gained so much importance is that now we have too much data and we don't know what to do with it. Now you can only do so much with certain databases or Excel. So there was a time when people used to use Excel to do data analysis. These days, not possible. So here's a quick view of how much the healthcare data has grown over the years. Now this was just 2013, which is basically yesterday. But since 2013 to as of now, you can see the growth, the immense growth of healthcare data. Like I said, this healthcare data combines a lot of things because again, we need the bigger picture to do any sort of analysis for drug discovery or any healthcare related analysis. Now, you might also, if you're new to omics data, you might have heard, so these are all buzzwords. What essentially is omic data? Omic comprises of these three things: genomic, genetically, so what is the genetic makeup of a patient, of a human being? That is genomic. Proteomic, protein-protein interactions, how do these happen? So these are some of the data points that we need to collect in order to get this bigger picture that I'm referring to. Metabolomics also, not entirely there, but it is absolutely essential to get that bigger picture of a patient and then use that information to treat the patient. Now, all this combined is what we have as omics data, and which is also just a tiny, tiny, tiny part of this entire bigger healthcare clinical data. Now, the sensor data is actually quite fascinating. So a couple of years ago, before Novartis, I used to work for Thomson Reuters. I don't know if you are aware of this. Thomson Reuters also used to have this science wing, which was essentially working on utilizing sensor data. That was new back then. This was in 2013. Now, what is sensor data? So the watch that I have right now, this is collecting a lot of information. So it knows my heart rate, it knows how many steps I have, it knows when I stand up, it knows when I'm not breathing. So all that information is being collected somewhere, and someone is using it. Now, there's also a lot of issues with data collection. Some people like it, and some people don't. Now, for healthcare reasons, it is important that we collect this data, and with consent, use the data for the right purpose. So when I say sensor data here, I'm referring to the correct use of this healthcare data. So my heart rate, I want somebody to look at my heart and say, maybe, am I prone to having cardiovascular disease maybe 10 years from now? It's absolutely possible to predict something like that using certain variables, certain parameters. So this basically gives that sort of information for a patient. EMR data. Now, I know you must have heard about EMR data. This is jungle, because when I was introduced to EMR data the first time, I didn't know how to understand it. So every time we go to a physician, every time for a general yearly annual checkup, certain things are collected. Now, these things you may think are just, okay, what medication, what's your height, what's your weight, they check your vitals. But all of this information, when you look at it from a data scientist perspective, this is humongous. So as a patient, I may not realize how big this is, but as a data scientist, I know how much effort goes into analyzing that kind of data. Forget analyzing, just how much effort goes in getting that data so that it's analysis ready. Now, the first step is source data. Where exactly is that all that information that you give to your physician stored? Does anyone know? EMR data? Okay. So now there are several different databases that have EMR data. Has anyone heard of Epic system? Great, of course you have. So the Epic is one of, Cerna is another. So there are a lot of these things. Now, between Epic and other systems, there are a lot of differences. So combining data, I would be really excited to know how your company does that. Combining these different kinds of data sources together is a humongous job. So that's something that takes data scientists more time than actual analysis of the data. So if somebody is giving you the data, great. Now, once you have the source data, like I was saying, data extraction. So now these are in very, very various different formats. Now, you might know when you go to a physician, most physicians these days have a laptop open and they're typing something. So you say you have a fever, the doctor isn't looking at you, but the physician is actually typing on their laptop. The reason that they're doing that is they're capturing every little thing. Now, in the olden days, let's say maybe 15 years ago, or maybe even 10 years ago, that wasn't the case. 15 years ago, the doctor would come in, look at you, you would tell them whatever you were feeling that day, and they would actually write it down on a piece of paper. Now, how do you get that information from that piece of paper that your physician has written down? How do you pass that information to, let's say, your next physician? Let's say you want to go to a cardiologist and your PCP wrote something down on a piece of paper. How does your cardiologist know what the PCP wrote down? Now, that is very critical information. How do you get that information all the way to your cardiologist so that they know what medication to prescribe? That's where you might have heard natural language processing came in. Has anyone heard of NLP? Great. Great. So natural language processing, essentially, for people who haven't heard of it, is extracting this information from that piece of paper. So someone has written something down. You are extracting that text. You are extracting those words, those characters, and then you're making sense of it. Now, this NLP is its own field. I mean, it's a huge field because obviously it takes a lot of effort to get that information from that piece of paper to a tabular format so that that information can then be analyzed. So there are multiple steps before you get to analysis, and then from that analysis, from those results is when you go to the treatment. So from getting the information to treatment, there are so many steps, and which is what I will be covering today. For EMR data, there's one, yes please.
Before you go too far, I just wanted to ask you, with some of the more consumer-oriented devices like the watch and et cetera, how do you ensure data integrity? So, for example, there's so much variability in capturing heart rate on your watch
Yep.
that you don't really know if the data is
Accurate or
reliable.
not, yep.
And then with the EMR and the source data, I think as probably many people have probably experienced, you go to one hospital, they collect one set of information, then you go to another hospital, and that information hasn't transferred, even within systems, like UMass, for example, they don't transfer. And so then there is a loss of data that occurs from one entity to the other. And then, of course, the patient is not always mindful of, I think, what they're taking. So they don't remember the dose that they're on, they don't remember the frequency. So all of this data error that's going in, how are you correcting for it, I guess, and ensuring that there isn't all this garbage in, garbage out?
Okay, so I'll answer both of those questions, which basically have very similar answers. To begin with, there is no one exact answer to that question because there really is no one solution to it. How do you correct the error? But there are certain things that you can do to mitigate these errors. I mean, not completely remove them, but at least minimize them a little bit. So one way that, from my experience, that I have seen is the amount of data that you collect. For example, you gave a great, heart rate. So the amount of data that you collect, just for me, let's say if you collect my heart rate every minute versus, let's say, every 2 hours, difference. So if you are collecting my heart rate every minute, you have that amount of data available for you. So it's easier to look at outliers, for example. That's a big one, because if my heart rate all of a sudden drops to 20, when, let's say, average is 120, I'm just giving you a number, something's not right. So that is one of the, I think, easier ways to deal with that issue. With EMR, because good thing that you brought it up, because I went through the same kind of situation a few years ago now. So you're absolutely right. Even within hospitals, data doesn't get transferred. But these days, I have to say, fortunately, that has improved quite a bit. So previously, like I said, piece of paper would be lost, or nobody would, put it somewhere. The files would be gone. So somebody had to go back, hunt for these pieces of paper, try to figure out what it was. These days, because the physicians are actually doing a great job typing everything in, it's a lot easier to get that information. But that doesn't mean there isn't any error. Like you said, patient themselves. So for example, if me, I'll take myself as an example. So there are all these, I don't know if you guys know of these apps, healthcare apps that, Google has one, I think Microsoft Health also has one, where you can answer a few questions, and based on those questions, it sort of gives you a diagnosis. I mean, they do say take it with a grain of salt, and by the way, which you should, but there are ways of how you can ask these questions. So I'll give you an example from Alzheimer's disease. Now, I used to, a couple of years ago, I was working on this Alzheimer's project. So there is something called quality of life. Has anyone taken or is aware of quality of life questionnaire? Great. Now, if you look at that quality of life questionnaire and if you try to answer these questions, so I may get 40 questions, but someone else might get 140 questions. Does anyone know why that is the case? Or that could be the case? Okay. The clear answer here is, if I am answering something and it looks, the collection, when the data is being collected, it looks somewhat consistent, it stops asking you questions. Let's say I'll give you 3 questions. One of the Alzheimer's patients have tremors. One of the questions could be, can you lift this pen? Let's say I say yes. And then next question comes up, it says, can you lift this paper? And I say no. There is an inconsistency right there. So now the data collecting machine, I'll just call it a machine at this point, the database, it knows something's not right. If a patient is able to pick this up but not this up, something's not right. Either the patient isn't answering right or something's up. That is when I get 140 questions where Raksha might only get 40 because she's being honest or she's answering the questions right. So like I said, going back to your questions, these are some of the things that have been employed to minimize error, but there is a very, very long way to go before we can say with certainty that there is no error. And I have to say, maybe there will never come such a time where we can say there is absolutely no error. But that is the state of AI machine learning. It's not even AI machine learning, it's just data collection at this point. Because what we have to understand is that it's not artificial intelligence or machine learning that we're trying to judge. It's the data collection. Your algorithm is only as good as your data. So if we can achieve a situation where we are absolutely collecting good data, then your algorithms are great. I mean, you can predict whatever you want to predict. You can do anything you want. But it's all about the data. Does that answer your question? Perfect. Any other questions? All right. Now, like I said, there's data mapping, and then you finally export this EMR data. Now, when you export this EMR data, there are different formats that this can be exported into. Some of these are tabular. Like I said, if something's written on a piece of paper, you again employ other artificial intelligence approaches like NLP, and then you extract that information so that you have this entire EMR data for a patient. Now, going back to this is only a small little blob that, that's part of this bigger picture of a patient profile.
What are the most common ontologies used besides?
From my thing, Loink and CDISC, Loink for lab codes, I mean, essentially, you have CDISC codes for all these disease domains. But from my thing, my experience, these are the most common ones used.
Not SNOMED?
SNOMED also for disease ontologies. I forgot to put that there, but absolutely. RX norm for, absolutely, I only gave 3, but RX norm for medication, SNOMED for any disease, hierarchies, absolutely. Imaging data. So I put this as last because I do have a use case that I would like to share with you guys. Imaging data is another big monster. The rest of the data, clinical data, can be in tabular forms, but getting this imaging data into tabular form takes a lot of time. Every time we go to a doctor, we get CT scans or X-rays or MRIs. All of these are just images, and there's very few information that you can just get on in tabular form. So you have, and the other thing is, let's say I get an MRI at Harvard Medical Hospital, and then my friend gets another MRI somewhere else. Those 2 MRIs are not the same because every MRI machine does things differently. Now, combining all this and making sense of all this and normalizing this information takes way longer time than actually analyzing this data. So imaging data, normalization, feature extractions. Once you normalize this, you can extract the features. Now, what features am I talking about? Let's say you have So the example I have is colon cancer example. In that colon cancer, you'll see that in a minute, but let's take another example. Let's take MRI. So I get a brain scan. What is it that I can see in that picture? Maybe I have a tumor somewhere. So how exactly is the physician seeing that tumor? Right? The pixelization, it's lighter color or darker color. The area around it is of a different color. So these are some of the features that you have to extract to make sure how big the tumor is or how dense the tumor is. How do you know how dense the tumor is? How do you gauge the porosity of that tumor? So these are all the features that you need to extract before you make any clinical decision from a piece of two-dimensional image. So that is the challenge that we see these days with imaging data. How do we get that from a two-dimensional image? And then classification is a machine learning, let's say, algorithm where you can say yes cancer, no cancer. That's a binary classifier. So how do you get to that? You need to do these three before you get to the classification step. Now the use case that I have, before that, data integration, there you go. But this I think we have already discussed. Data integration only happens after you have collected all the other information that I just mentioned. So EMR data, imaging data, omics data, genomic, proteomic, metabolomic data, as well as just plain old simple clinical trial data. Now, how many of you work on clinical trials? Okay, 1, 2. So when you talk about clinical trial, there are most of these could be tabular information. So you have like a patient ID and you have a bunch of rows, so it's basically an Excel format. So that what I consider to be an easy peasy way of looking at data. Because looking at all the other things, I think clinical trial data is a lot easier. So once you have all that information, clinical trial, omics, all of those, imaging and EMR, that needs to go into some database where a scientist like me can go get that information and then analyze the information. So that goes to a database where it can be queried. Before that goes into database, you have to process it. How do you know me, patient? It's the same patient who has imaging data or who has EMR data and who has a clinical trial data. How do you map patients? So that's another big thing that you need to do before you get to the analysis step. So building that database also takes a lot longer. So the reason why I'm focusing so much on just getting the data, integrating the data, is so that we understand that artificial intelligence and machine learning is just a tool. It's not magic. It's not something that can give you what you want. It can, and you can use it very well, only if you have all this information good to go. Clean information, clean database, clean data, and then you can apply your algorithm any way you want to. So challenges, I think I've already mentioned, syncing all these different data sources. So we discussed EMR data, where we have to sync from different kinds of EMR, like Cerna and Epic, those were the examples. Getting data into this big data structure. So we keep hearing this term big data, big data. What exactly is this big data that we talk about, especially in healthcare? Big data essentially means a combination of all of these things that I just mentioned in a big, let's say, Excel file. That is what we consider big data. The other biggest challenge we have is extracting the relevant information from this big data. How do we know that, let's say you're collecting some information, me as a patient, how do you know that is relevant? How do you know that information will lead you into diagnosing something? Let's say I have a heart attack or heart disease or something. How do you know? Can you just get that information from the heartbeat that you have here? Do you need other information? Do you need blood work? What exactly is needed to get that? And how are you making sure you are getting that relevant information? So these are challenges that we see today with healthcare data. All right, now comes the fun part. Once you're done with all of this data collection, making sure the integrity of data is good, that's when you can apply these three things. So I just put knowledge graph there because that's also one of these buzzwords that go on these days. Has anyone heard of knowledge graph? 2, 3 people? Great. So we'll just start with artificial intelligence. What here in this room, what do people think of when I say artificial intelligence? What is it? Anyone? Anyone wants to just take a guess? No? Good, because even I don't know. So the thing is, with artificial intelligence, people have various definitions. Some people, like let's say Google, is using artificial intelligence to do very different kind of things. So is Microsoft, but so is Novartis now. But what a pharma company would consider artificial intelligence may not be what Microsoft or Google may consider. Now, Facebook is collecting all the information that all the pictures that we post on Facebook. Facebook is collecting all that information. I don't know if you guys have noticed, if you, let's say on Amazon, you were looking for something, a purse. The next day, or maybe even in a couple of hours, you'll see that ad showing up on your Facebook. Has anyone wondered why that happens? Exactly. So data is being collected no matter what you do, wherever you do. A simple thing like Facebook, although that is not simple, it's collecting a lot of information. So is Google. Let's say you Googled something. You Googled a specific gene, or you Googled a specific heart condition, or let's say colon cancer. You will see an ad for colorectal examination or something at some point. It's going to happen. So artificial intelligence has a lot of meaning, going back to that point. For us, for a pharma company, artificial intelligence could just mean natural language processing, because that is where pharma companies are at the moment, right? So what is relevant to us is what that physician writes on that piece of paper. That is what we need. Right now, at this given moment, we don't really care so much about what the patient is typing on their Facebook. Although, I have to say, at some point, that is going to be very, very useful, because you might be surprised at how much information we give out on Facebook or other mediums like that, or Instagram or something. So if I have a stomach pain, maybe I just say, hey, Chelsea, I have a stomach pain. Guess what? That information is being collected. Now, Facebook knows that you have a stomachache, and you will get an ad for that. So that's artificial intelligence. Machine learning. Any guesses on what machine learning is? Okay, I will give you the answer. So machine learning, like the name, is basically how you teach a machine. That is all it is. How are you teaching a computer? That is essentially what machine learning means. In machine learning, let's say we have this laptop. I have a laptop. So how do I teach that laptop that does not have a brain like us? How do we teach that thing to distinguish between, let's say, a cat and a dog? So there is something called labeled data. Does anyone know what that means? No? Okay. Labeled data essentially means you have a certain condition. I'll give you an example of oncology. So I'm a patient. I have a heart rate of 20, but let's say I have a heart rate of 20, and I have some blood report which says something's not right, blah, 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 and then I have been diagnosed with colon cancer. Now that is my report. So what I tell the machine is, hey, this patient, Shruthi, has this heart rate, this blood work, and she has been diagnosed with colon cancer. Now I've labeled patient, me, with colon cancer. Now that is labeled, right? So that is supervised. Now what is unsupervised learning? Exactly the opposite. Now I give my laptop a set of information. It is an Excel sheet, let's say a tabular format, but I don't know what I have. I have all the other information, but just that diagnosis column is missing. So there's blood work, there's genomic data, there's imaging data, there's all of that data, but that diagnosis does not exist. And now I'm asking my laptop or my machine to tell me, hey, do I have cancer or not? That is what we call as unsupervised learning, machine learning. Knowledge graph. Anyone has heard at all buzzword? No? Okay. So knowledge graph is a very funny, I have to say it's quite funny. So a knowledge graph essentially is a database. So you guys know what a database is, right? So it's basically a big database. It's a warehouse where you can input all this information in a tabular format. Knowledge graph is also a database, but it has a little kick to it. The reason why it's a little different is because it can incorporate several different kinds of information. So I was saying there is imaging, there is EMR, there is some other thing. Let's say all these have not been mapped to a single patient. So it's not in a tabular format. So I'll just use that for a change because I'm bored of this. So let's say there's EMR here, and then there's imaging here. Now, in a traditional database, this will be like, let's say, patient ID, EMR information, let's say age, and from imaging you're getting some diagnosis, right? This is what a traditional database would look like. Now, it's unlikely that you'll get all this information that cleanly so that you can use this. This is dream come true for data scientists. What usually happens is you have EMR, you have imaging, you have some other data that you don't know, and there's NLP data that's coming from some other thing. So there's a lot of different things that are just sort of bombarded at you. So as a data scientist, what do you do when you see something like that? You say, okay, now I don't know what to do. But that's when knowledge graph comes in. Knowledge graph, essentially what it does is it sort of draws connection like that to each and every blob. So knowledge graph sort of creates these two-way associations where you can say, okay, this patient, EMR, sort of looks like this patient who also has imaging that corresponds to this patient who has this doctor's note. So it's drawing this sort of not conclusion, but just associations between these blobs. So this is not conclusive. It's not that you can say, oh, knowledge graph, it just did blah, da, 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 and there you go, that's the patient. No. But what it does is it sort of gives you an inclination. It sort of gives you a direction in seeing, okay, maybe this patient, this patient, this patient, this patient are one and the same. Maybe there is some connection there. Maybe patients who have this specific kind of blood work also seem to have this specific kind of imaging data, also seem to have this specific kind of doctor's note. So it's basically just drawing those associations. Now that knowledge graph is an excellent tool, but like I said, it's always better to cross-check. So we're not in a position where we can leave things to machines. We still need humans, which is where we come in. That is what it is. Questions so far?
Some questions. So what's it helping you now? Is now the undiagnosed patients? Diagnosed patients? Because you say, okay, there's a lot of data from diagnosed patients. So you say, hey, we run into a database and we find people that are undiagnosed.
Could happen.
Yeah, because I'm wondering, because in a controlled setting like a clinical trial,
You will have
complete control what you get. So I'm not worried, I'm not so sure if this would be beneficial to a clinical trial.
So it doesn't
Unless you have undiagnosed patients that you're looking for.
Correct. But so unsupervised doesn't necessarily have to be just for diagnosis. Let's say you have diagnosis information, but you want to find something else. So the example I just gave was, if I want to predict if someone is diagnosed with colon cancer, that's easy because you have that information in a clinical trial data set. But let's say you need to predict something else. Let's say you have diagnosis of colon cancer, but you want to see, or let's say IBD, because IBD usually progresses to colon cancer. Let's say we have an IBD clinical trial data set where we don't know if a patient is diagnosed with colon cancer, but you wanted to predict if that's possible using rest of the data. Now that would be an unsupervised learning because you are trying to predict something that does not exist in that data set. Does that answer your question? So this knowledge graph is not for diagnosis, or it's not for predicting. Knowledge graph is just a database. What it's doing is, with those blobs, it's sort of budging you into creating something like this. So it's doing this, so that you can create this data file. But from this data file is where you get to do the actual analysis and predict and do all those funny things. Does that answer your question? Any other questions?
Two-part questions. Does not make for semantics, like what are you actually connecting? Is the connection, does data tokenization have to do it?
So I didn't hear the last part.
The data tokenization part to connect the databases.
So the first is semantics. So does it include?
Semantics of what you are actually connecting. Say, does it have a language associated with that to actually say this is an image?
Yes. The answer is yes. So it does. So there is a, so I won't call it normalization, but there's absolutely a translation. So everything that you're collecting into that knowledge graph database needs to go through this. So there is a, so it's very similar to a query language if you have done any database query. So that language, the syntax looks very similar to that, but there's a terminology, there's ontologies that basically help the user to incorporate all that data. Now, second part of question was?
So just to follow on to that, is that semantic networks 2.0 again, or is this something completely different?
You have multiple ways of doing it, but that could also be one of them. So what I've used, or from my experience with knowledge graph, I have not used that. But there is, so there's that theory exists, so that absolutely you need something to incorporate all this information, otherwise it doesn't work.
The second part question is the data tokenization, wherein when you have multiple databases, most of the recent technologies such as data landers can connect them using patient as the
mapping, yeah, correct.
And is that how it's done with
No.
the knowledge graph, or
It's different. So what you're saying you can do with just a simple R script, right? So let's say you have patient ID in EMR, patient ID in the imaging, and all these are tabular things. So this is an Excel file. Let's say this is an Excel file. So is this, so is this, so is this. And each one of those Excel files has a patient ID column. If that's the example, in that case, it's easy to connect everything, right? You can map.
so it will be from two institutions who cannot share the patient ID.
So just anonymous ID. Okay, in that case, you cannot use patient ID as the mapping. Absolutely. In that case, knowledge graph is useful because what essentially knowledge graph will do is it will look at other variables. So it'll try to, so there's some artificial intelligence in knowledge graph. So it sort of looks at these patterns. But that again is only good as long as, so the amount of data really has to be humongous for this to work. So you cannot do this with, let's say, with just one clinical trial data. So it'll be too little data to use something like this. So this is truly big data problem. But let's say you do have that much data, then this is something that you can use to create these patterns. So it will actually tell you, okay, this patient looks a lot similar to that patient in that database. So maybe draw a connection there or draw an association there. So that's the whole purpose of knowledge graph.
So just a question. So from my understanding, knowledge graph is a higher level of clustering kind of thing.
So I wouldn't.
So it's a multi-variable clustering.
I wouldn't call it clustering per se, as in machine learning clustering. So it uses similar theory, but all it's doing is it's sort of associating the patients or groups of patients like clustering. But when you use the word clustering, it essentially means it's putting these blobs into, yeah, it's not like it's not even close to that, which is why it's helping you figure out this big Excel file, but it is not doing that PCA, no.
Another question. Does that solve the problem of putting together multiple data from multiple centers, where you have multiple databases normalized differently between different centers, structured differently? Would that be solved by using this knowledge graph?
In theory, yes, but it has to do with how much data you're collecting. Let's say you're collecting 10 patient data from Institute 1, Institution 2, 10, 10, 10. No, it's not going to work. So you need to have enough data so that this can see that pattern. So it won't see that pattern, let's say even with 100 patients, it's not going to happen. So it has to be a lot more, and that pattern has to be between features and not just patients. So that is how it's associating, through features and not through patients. Makes sense, right? All right, any other questions?
Not so much questions, just
Yes.
just like a discussion point. So like in chemistry, so in research, looking at those three and then looking at knowledge graph, you'd probably tying together, like say previous chemotypes for certain drugs, and then looking for patterns, that all these compounds had, say, a methoxy group, and
You can.
kind of picking out.
So you can also just use NLP for that. So you don't even have to go as far as knowledge graph for that. Let's say so that's a great example. So oncology again, let's say colon cancer. So colon cancer, you have some clinical trial data, or you have these molecules or the synthetic molecules that you're creating, and you have some patterns where things have already been created, and you want to look at these patterns. So natural language processing essentially can read through these patterns. So these should be online, right? Or even if they are hard copy PDF files, natural language processing can basically go in. Let's say you are looking for molecule XYZ, right? I mean, let's say that's what you're looking for. So it can go through, read through each of these patterns, each of these publications, each of whatever you want it to read, and pick out that XYZ from everywhere. So that tells you, okay, this is XYZ molecule is what I'm working on. There's a pattern that also has XYZ in it. So you can make it as complicated as you would like, but the more complicated you make, the less results you will get. So that's essentially how it is. So does that answer your question? All right, use case. So this is a pretty picture that I sort of copied from my previous publication, but what you see here is colon cancer tissue. The top two are H&E stained. The bottom two are exactly the same tissues, but they have been remeasured using scanning electron microscopy. The difference you see here is these are decellularized, so it's just the extracellular matrix that you see here. Does anyone understand? Yeah, I mean, so it's just without the cells, decellularized extracellular matrix. So let me give you a little background on what I was trying to do when I was trying to get these images. Essentially, this is part of the pattern that I have, but what it is let's say I'm a patient, I have IBD, inflammatory bowel disease. Now, historically and in literature, there is a lot of connection between IBD with colon cancer. So with almost 20%, this was 10 years ago, there was a 20% chance that a patient with IBD will, in 10 years, develop colon cancer. Now, knowing that statistic back then, I was thinking, what if there is a way that you can look at tissue samples of IBD patients and then somehow try to predict how likely is this patient to develop colon cancer in 10 years? Is it there? Is it not there? And if it is, what grade of colon cancer? So that's essentially what my thesis was. That's essentially what the patent is. So going back to this. So one of the first things that I had to do was, if you have seen images, just tissue sample images, these are two-dimensional. What I was trying to get here was, within a tissue sample, does tissue porosity, does tissue features like the protrusions within the tissue sample, do they provide us information about the onset of colon cancer in patients with IBD? Now that was the broader question that I was trying to answer. What you see here is normal tissue. What I consider normal is non-carcinogenic or non-tumor tissue, which means these are all IBD patients. So the top picture that you see is of a normal non-colon cancer patient. The bottom is of a cancer patient. Now these images are two-dimensional. How do we get from the two-dimensional images to three-dimensional? Because that is essentially what I was looking for. Now, if a tissue is rough, like a lot of protrusions, how do I measure those protrusions? So you need to get that three-dimensional image. So that was my first sort of experience with image processing, as well as these days we call it artificial intelligence. Now, the way we did that was using the two-dimensional images. So we had about 500 patients, 500 normal, 500 colon cancer patients. For each of these patients, we sort of created this tilt So we programmed it in such a way that for every image, it would create a 10-degree tilt this way, 10-degree tilt that way. And that would basically get us to something that looks like this, 3-dimensional. Now, from that 3-dimensional data, we could extract the little protrusions. Now, it's important to know that these protrusions were in nanoscale, so tiny, tiny, tiny protrusions, but nonetheless, very extremely important information. Okay. Now, porosity also. So one of the big giveaways when I was looking at this was, look at the porosity. So porosity essentially is how quickly can cells penetrate through the extracellular matrix. So that's essentially what metastasis is. So if the porosity is almost, look at this, almost 3 times. So in a cancer patient, the extracellular matrix was about 60% porous, which means it was that much easy for the cells to just sort of go through. But in a normal, the cells just wouldn't go through, because that's just the ideal normal way of being, right? Now, based on that, I started digging deeper. So this is just method of how we do it, how we get the extracellular matrix, how we decellularize it, we remove the epithelial layer, and then that is what you see. This is the same image. So this basically becomes that. And that happens because you're introducing this 10-degree tilt and you're seeing what the protrusions are. Now, protrusions are, I don't know if you can see it clearly, but these are the tiny protrusions. This is like a dark circle right there is the how deep it is. So how deep something is or how high something is basically gave us a lot of information about the prediction of colon cancer. Now, from that image, we created something called a roughness profile. Right? So now in roughness profile, so it's basically telling us how high, how low in nanometer scale. Now using that, I was, for my PhD and just for the thesis, I was looking at these 4 features. Now there were a lot more features that we could look at, but since it was a PhD thesis, there was limited time, so this is what we focused on, because based on literature, we thought these were the 4 most important features that would tell us more about this onset of colon cancer. Okay. Now, if you just plot this and compare normal versus carcinoma, and this is roughness profile, average roughness profile over 500 patients, you see a drastic difference between what normal average looks like versus carcinoma. And this was like, this is fabulous, because if there is such a distinction between normal and carcinoma, there definitely is something that you can extract from this and use that information, relevant information, to diagnose the onset. That was the idea that we were going for. And this is just the same, just giving you the numbers in nanoscale. All right, now this is where, I'm sorry if the images aren't that clear, but essentially what I've used here is a support vector machine. It's a classifier. So I know some people already know what classifiers are. Support vector machine essentially is a supervised learning. So you sort of teach the machine. It's a machine learning technique. You teach the machine. So it's labeled data. So you have 500 patients that have these 4 parameters I just said, the roughness profile parameters, and you also have the diagnosis attached. So you'll say patient 1 has this roughness profile and has colon cancer. Patient 2 has this roughness profile but does not have cancer. So like that, we have 1,000 patients, 500 each. So we teach the machine, and machine learns from this data set. Once it learns, you introduce another set. So you introduce something that you don't know. So there is, the diagnosis column is missing. Now when you introduce, it can predict, it can tell you, it can classify just using the roughness profile if the patient is normal or has a tumor. So everything in red, I don't know if you can distinguish the colors, but everything in red falls, it's clustering again. So everything in red basically falls to one side, everything in green, normal, is on the other side. So the point of this is that it's using the information, extracted information from images, we were able to classify patients, diagnose patients as either they have tumor or they don't have a tumor. So for today's presentation, I will end here, but this continues. So this isn't the end. Just classification is just one machine learning algorithm. Artificial intelligence, machine learning, the whole purpose of that is prediction. So you should be able to predict something based on the humongous amount of data that you have. So we did do that, but in this presentation, I think we're almost running out of time. But yeah, so basically that is what we did. So I will stop here and just see if anyone has any questions. Yes?
Can you just take like a break 2 minutes? Just how did you predict it after you diagnosed?
Okay. So there is an entire sort of an image, but so the way we did it was after that classification that you see here, once we have all that information, we also looked at different grades. So for each grade of colon cancer, there were very specific roughness profile that we could generate. So even between, let's say, grade 1, stage 1, all these different levels, we could see in nanometer scale, we could see a lot of difference. So for each of those, similar concept was used, similar roughness profiles were generated. After that, what we did was we used not classifiers, but we used prediction. Sorry? Exactly. We used regression algorithms just to see how well can this predict. And based on the classification, since the data was so easily separable, we were able to predict with 95% accuracy. Now, for something like that, I have to say we only used 4 variables. So let's say if we had used 20 variables, maybe the outcome would be a little different. But with those 4 variables, the prediction rate was about 95%. So that's basically what we tried to do. Yeah. Alrighty.
How is data then used to help the patients? Were they communicated with the patient and physicians, and they did some kind of preventive?
So this was just a patent. So we are still trying to commercialize this. So this I did as a PhD student, so I graduated, but someone else sort of tried to commercialize this. So this is still, I graduated from University of Florida, so this still belongs to University of Florida. But we could not directly impact patient lives using this because this was more of an experiment. This was more of a thesis that I was working on. But the idea of this was, yes, it can be used. There's nothing stopping us from using that. But again, it has to go through jump a lot of hoops before we get there.
Right, so you did that in 2012. So did the University of Florida do a follow-up to see how many of these patients did develop Alzheimer's?
Yes. So there was a follow-up study that was published, but it was a clinical paper that was published. And what they said was with 95% accuracy, a lot of these people, it wasn't 95% accurate, but there were. So what we sort of diagnosed or predicted did come true. But it's a different publication. I'm not the author on that one, so I did not put it there. But it absolutely exists.
Were you able to correlate that with time?
No. So we did a 10-year sort of a study. So even now, so that 10-year isn't over. So it's not that the study is complete, but this algorithm basically said that with 95% accuracy, we can predict over a 10-year time frame that these patients will develop colon cancer with 95% accuracy. So we cannot still answer that question with certainty because 10 years hasn't happened, but even before that, 4 years after this was published, there was a follow-up. But in that 4 years, we already saw certain patients who developed colon cancer. Yeah. All right, any other questions? Okay. All righty, so I think that is basically the conclusion that I have for you, for this presentation. But one thing I do want to say is artificial intelligence, machine learning is extremely cool. It's fascinating. It's unbelievably, interesting to see how you can use these different approaches. But one thing we do have to keep very much in mind that it does not imply that it can cure everything. We have to understand that, yes, it's usable, yes, it's great, but there are a lot of caveats that go with using AI and machine learning tools and all of these things. So All right, hopefully I haven't discouraged people too much from getting into the AI machine learning side of things. I mean, honestly, I really do enjoy working on this, but yeah. But do look more into it, learn more, read more. There's a lot of things coming up every single day. You see something new. Knowledge graph especially is something new. So you probably hear blockchain and this and that. What we also have to understand is none of this is new, and none of this is that groundbreaking. It's all, if you think about it, it's all common sense. But common sense, when applied to the relevant thing that you do, becomes magic. And I think that's what the pharmaceutical industry right now is trying to do. Use what's available and get something out of it. Create impact. All right, that's pretty much it. Thank you.